So, hi everybody. My name is Justine Karst, and we are here today in the What We Are Reading YouTube channel of the South American Mycorrhizal Research Network. Today, I'm really excited to interview Dr. José Medina-Vega. So, Jose is the lead author on a paper that came out recently in Nature Ecology and Evolution. The title of it is Tropical Tree Ectomycorrhizas are distributed independently of soil nutrients. Jose, do you want to introduce yourself and just tell us a little bit about yourself?
Hi everyone. My name is José Medina-Vega. Yes, I'm originally from Ecuador, but I'm currently based here on Washington D.C. on the Museum of Natural History. I'm a postdoc at the Forest Geo Network, which is part of the Smithsonian Institution. I'm happy to be here.
Thank you. Okay, so we'll jump right into the paper. I have a few questions I want to ask you, and I'm excited to hear your answers. Okay, so just to set the stage, can you please explain the topic your paper addresses and give us a sense of what was the thinking on this topic before your study came out?
So our manuscript examines the distribution of ectomycorrhizal trees in lowland tropical forests. So there is this prevalent hypothesis in ecology, and some people refer to it as the Reed's rule. The main idea is that the abundance or the distribution of ectomycorrhizal trees will decrease from high latitudes and high elevations to low latitudes and low elevations. And this is because in areas at high latitudes and high elevations, you have slow decomposition rates. So slow decomposition rates leads to low availability of nutrients in mineral form. And the idea is that ectomycorrhizal fungi is better capable, it's more capable than arbuscular mycorrhizal fungi in mobilizing these organic forms of nutrients in the litter, right? So it takes the nutrients from the litter and sends it to the tree. So in lowland tropics or at low elevations, you have faster decomposition rates because the weather is warmer and it's wet. So this is not really an advantage there because nutrients are readily available for the plant. So in lowland tropical forests, the idea is that you won't find ectomycorrhizal trees, or well, they are rare. They call them rare. And if you find them, you only find them in monodominant patches and in patches that are on forests with very low soil nutrient availability. But when you look, so this generalization does not really align with observations from the field. So for example, if you look at Southeast Asia, in Malaysia and Borneo, that's an area that we know quite a bit in forest geo, you will see forests that about 40% of the basal area is dominated by ectomycorrhizal trees, which represent about 10% of the species. So there is, and you see like a gradient in the abundance of ectomycorrhizal trees. And it's not that they are only in areas with low soil fertility. These forests are not nutrient poor forests. So seeing this connection between the hypothesis and what we see in the field, we decided to write this manuscript.
Yeah. Okay, yeah, so one thing I really liked about this paper, there's many things, but just the data set and the locations, like I feel like we have so little information about mycorrhizas and their trees in that part of the world. So that's really one of the things that caught my attention. And kind of, I think is what you alluded to, is a lot of our thinking comes from, say, North America, Europe, those kinds of places where we've developed these generalizations that might not hold true everywhere else. Okay, so how then did your study challenge these ideas? And I guess and that'll be in combination with my next question is, walk us through your approach. We've got the setting, the background now. So how did you challenge these ideas? This Reed's rule, I guess.
Yeah. So the manuscript challenged two main ideas. One is that ectomycorrhizal trees are not rare in tropical forests. You can find them in many places, particularly in Africa and in Southeast Asia. In the neotropics, it's a different story. It has to do more with biography, not with low soil nutrient availability. But in tropical Africa and Southeast Asia, you can find them like anywhere. That's the first idea that it challenged. And the second idea is that the distribution of these ectomycorrhizal trees, wherever they occur, is actually not related to the availability of soil nutrients in mineral form. So you can find areas with high or low abundance of ectomycorrhizal trees in areas with high and low soil fertility. So these two things are disconnected, the way that we are measuring, the way that we measure things, right? So our approach was basically, so we compiled two data sets, one that we call a coarse scale data set and a fine scale data set. The coarse scale data set, which it comprises 31 plots, it uses basically the mean abundance of ectomycorrhizal trees, plot level mean abundance, and the mean soil nutrient availability. The fine scale data set has 16 plots. Each plot is about 50 hectares. So each plot we divided by 20 by 20 meter quadrats. In each of these quadrats, we quantified the basal area of ectomycorrhizal trees and the soil nutrient availability for that quadrant. So we have a fine scale. We can quantify the fine scale association between the relative abundance of ectomycorrhizal trees and soil nutrient availability. We only had 16 plots. That was 16 plots only the fine scale data set. For the 31 plots, we use mean plot level data because we didn't have that detail of information for the other plots. But we wanted to use most available data that we had.
And the 31 plots, like, so these are scattered in lowland tropical forests. So Africa, South America, Southeast Asia, in that kind of belt, right? Across the world?
Yeah, exactly. And the 16 plots too. So we have from the 16 plots, the fine scale data, we had 5 in the Neotropics, 9 in Southeast Asia, and the rest in Africa. So they cover a large part of the lowland tropics, right? So then we had the abundance of ectomycorrhizal trees and the soil nutrient availability at these two scales. And then just using regressions, we check whether the distribution of the ectomycorrhizal trees was related to the availability of nutrients in mineral form. Okay. That was our approach.
Yeah, yeah. Okay, thank you. So kind of on that topic, okay, so you argue that the relative abundance of ectomycorrhizal trees is unrelated to variation in the soil properties that you measure at both these fine and coarse scales. So one question I always like to ask everybody is, are there alternative explanations for your results? So you're saying that they are independent of soil properties, but is there anything going on or kind of any lingering, like, oh, we should also check this, or other factors going on that might explain these results?
Yeah, there are a couple. There are a couple. And I will mention 3 that I think are key.
Okay.
So the first one, and you touched on this earlier, you mentioned that we assume that fungi in the lowland tropics will perform similar to what they do in temperate and boreal regions. Okay, so let's consider that possibility. Let's assume that it works that way. Okay. So ectomycorrhiza fungi in lowland tropics having a higher capacity to mobilize organic forms of nutrients. Okay, that's one thing. That's one thing.
Yeah.
Another thing relates to how much of those nutrients actually the fungi transfer to the host, right? There are a couple of studies that show that the exchange of carbon and nutrients between the host and the fungi is not universally reciprocal. You will have cases where the fungi are actually hoard nutrients because they also need nutrients for growth. And well, you will probably see this in areas with low soil fertility. And if this happens, well, that will disconnect any expected association that you may want to see. So that's assuming that they will function similarly. Okay, now let's step back. So how feasible is that? Well, how likely is that? We know that, okay, evolutionarily, ectomycorrhizal fungi are more recent than arbusculomycorrhizal fungi. But they are more diverse. So they have like about 20,000 species, but arbusculomycorrhizal fungi about 345 species. So assuming that all these 20,000 species will perform similarly wherever they occur is highly unlikely. And in fact, there are several studies that show that ectomycorrhizal fungi commonly occur in areas with high soil fertility or low soil fertility, independent of the host identity or the host distribution, probably with different costs and benefits for the host plants. So that's the second point. The third point will be the data that we have at hand, like the the abundance of ectomycorrhiza trees and the fine scale soil fertility, this is not only for this study, this conflicts with me when we talk about soil fertility. Are we actually, when we talk about soil fertility, what do we mean with soil fertility? Do we actually mean what the plants, do we know what the plants,
How they
do we understand?
perceive soil fertility?
Exactly.
Yeah,
So
yeah.
that's one thing that we're measuring. We have RAP, we can use Olsen, we can use resin tapes to get full fluxes of nutrients, but it's actually what the plant perceives. Are we actually measuring what we're supposed to be measuring? That can be another explanation. Probably we're not really capturing what the plant is actually needing. We're talking about many species. We have plots with thousands of species. We have other plots with 300 species. And assuming that it's the same fertility gradient will be filled by all the species in the same way, that's probably unlikely. We don't know.
No, that's a really good point, Jose. For the first time this past fall, I visited Panama, the Stry Station there, and we walked through the forest. And so all of my research has been done in the Boreal forest. And what blew me away was how many species. And I mean, I know, I've been taught this over and over again, like the high diversity in the tropics, but it really didn't resonate with me until I walked through that forest and just realized how many species were there, and what a challenge it is to figure out, how is this system functioning? I mean, I find it challenging to figure out how a boreal forest functions, and it's, a complex forest, but then those forests down there, yeah. So I think that's a really good point. And then thinking about, what we measure, is this actually what a tree or a fungus actually experiences? And I really appreciate your point too about the high diversity in ectomycorrhiza fungi. I think we often do sort of categorize AM and EM fungi into these very coarse groups, but there's so much diversity, I would say, especially with ectomycorrhiza fungi. They can tolerate, a spectrum of nitrogen conditions. And I think sometimes it's sort of easy to forget that when we're really looking for ways just to generalize the system. So definitely, I take your points. Okay, so kind of on this topic, so you mentioned Reed's rule. The other framework, though, that your study intersects with is the main framework. So for our audience, so this is the Mineral Associated Nutrient Economy framework that Rich Phillips developed. So this framework came out of temperate forests in Northeast US. And so obviously the it works really well for those forests. But it doesn't seem, I think you would argue that it doesn't work for these tropical lowland forests. So any ideas why it works so well in one forest and not in another forest?
Yeah, this framework is really interesting, and it seems to work well in temperate forests. There are very few cases in lowland tropics, not lowland tropics, in the tropical forests where this framework seems to work. But these are exceptional cases. This will be in areas that have, such as monodominant stands, where you have, for most of the stand is composed of 1 or 2 ectomycorrhizal tree species, or in very unfertile soils in the dry tropics. In fact, a manuscript from Rick Phillips' lab, I think, in 2018 from New Phytologies, where they compare litter decay rates between arbuscular and ectomycorrhizal trees globally. Yeah. So they do show that the composition rates of ectomycorrhiza tree litter, the composition is lower than arbuscular mycorrhiza tree litter only for temperate systems, right? But in subtropical and tropical systems, both the litter of ectomycorrhiza and arbuscular mycorrhizal trees, they compose very fast. So regardless of the initial conditions of the litter, they will decompose really fast, driven by the hot climate, wet climate, right? So if litter decays do not really differ, it shouldn't be that surprising that the nutrient cycle isn't different between arbuscular or ectomycorrhizal dominated stands. So as this litter decomposition often establishes the conditions for biochemical dynamics in the upper soil layers, right? So if you have, regardless of what your initial condition with the litter is, if both decompose really fast, so there won't be any difference in the nutrient dynamics in those systems. That's what I think. But in temperate forests, because ectomycorrhizal litter decomposes slower, and it seems to be consistent with the many studies that showing that. So you expect to see that difference there.
Yeah, yeah. No, that makes sense. So in your, I'm going to call it your region, in the locations where you studied, so the ectomycorrhizal trees, so their distribution and abundance not correlated with the soil properties you measured. What then, why are they where they are? Do you have any ideas on that? I know, and
Yeah,
that's a that's a that's a big question, but so just
yeah,
yeah,
it's like why dipterocarps are not in Africa or
yeah,
Detaroileum are dominant in Africa but not in Southeast Asia. Yeah,
yeah,
we made a point in the manuscript. I don't really know the answer to that question. That's probably related to another field, but
yeah, yeah. No, it's definitely. I mean, these are I probably like evolutionary, geological, big time and spatial scales, but that's definitely what I was left wondering at the end of the paper. Just sort of this other question of like, why, like, why are they there and not somewhere else? And
yeah.
but
And
yeah,
why their abundance is really low in the nootropics.
yeah, yeah. Yeah, and
Yes.
I mean, and to be honest, I was really surprised, and I think I had this stereotype of tropical forests as well as being very uh, low abundance of ectomycorrhizal trees, and so it was good for me to read this paper to kind of update my knowledge on that region. Okay, it's kind of on the next steps. So, what do you think is the next steps in this research?
Well, as the next step, I believe that one of the first priorities should be to gain a deeper understanding of the functional biology of the ectomycorrhizal fungi, right?
Yes,
That's key.
I totally agree. Yeah.
Yeah, it is very unlikely that given their huge diversity, they will do the same. Yeah, it is very unlikely. I mean, let's look at their hosts. Let's look at the host, for example. We know that well-known ectomycorrhizal host lineages, they occur in a wide range of soil and hydrological conditions. For example, the genus Cocolobal, which is from mostly abundant in the nootropics. You can find it in many places in the nootropics, regardless of soil conditions, right? Eucalyptus in other areas, same. Dipterocarp in Southeast Asia. For instance, within the dipterocarp, you have the genus Shorea, which has more than 100 species. And when you go to this forest, you will see that many species within that genus, some species are restricted to areas with high soil fertility. Other species are restricted to areas with low soil fertility within the dipterocarps. So we know that the ectomycorrhizal host lineages are very adaptable. So ectomycorrhizal fungi should also be adaptable. I mean, some studies show that they can be found in areas with high fertility and low soil fertility. So I think that we first need to stop assuming that they will do the same thing. Then gain to a big deeper knowledge of what they're actually doing, understand a bit more to be able to make big approximations of what is driving actually the distribution in the at the
Yeah.
pantropical level and global level.
Yeah. Well, and I wonder too if part of those generalizations will be that things work differently depending on where you are. And, and that might be one of the take-home messages is that, say, how the boreal forest functions doesn't necessarily it will not predict how a lowland tropical forest functions, even though ectomycorrhizas and trees are in both places. I mean, it seems very obvious, but again, I think part of the issue, though, is that where these ideas have developed are in very different forest types than, say, where you're working in. And so, yeah, I really appreciate your work on this topic. Okay, on that note, I noticed, well, when people look at the paper, so there's a lot of co-authors on this paper. There's over 30 people that you worked with. Any tips for the audience and how you write a paper with over 30 co-authors? Was it a smooth process or was it difficult?
It was a smooth process, but it took some time.
Yeah.
Because you need approval from all co-authors, right?
Yes.
Uh, you need to consider their ideas. In several cases, you cannot incorporate everybody's ideas. In some cases, because they will contradict each other. In some cases, they will align to each other. At the end, you make the final decision. That's a point that needs to be clear from the beginning. But everybody needs to be on the same boat, right?
Mm.
So in terms of, but one thing that I found important, and I read this in a paper, I don't remember who was the author, is that if you send a Word document or a Google Doc to all these co-authors, everybody will, it will be very problematic to work with this file. So what I did, I read it somewhere. You send a PDF and a Google spreadsheet with lines on the PDF, and everybody can comment on the Google spreadsheet. So that allows everybody to see what's
Oh,
everybody commenting on by
oh,
lines.
yeah.
If that comment is actually being taken into account, if that comment was already done by somebody else, and then you can keep track on time. So the first review, second review, third
Yeah.
review, this comment was already
Yeah.
included, not what's happening.
Yeah.
So
Okay.
that helped me a lot.
Yeah, I have not heard of that before. But that sounds fantastic. Wow. Okay. Well, thanks for sharing that.
Yeah.
Um, any concluding remarks, Jose? Any last thoughts about your research or
I think, no, I think we covered the key points, the key message of the manuscript. We also discussed a little bit the future, what needs
yeah,
to be done, what needs to happen. Very excited to see what's coming on now, next.
yeah.
Uh, yeah,
Okay. Well, thank you very much. And I want to say again, thank you for your tremendous work. The other thing I realized when I visited Panama was the climate, very, very hot and humid. And I just have such a new tremendous appreciation for people working in tropical forests. Yeah, so yeah, thank you for the fantastic work. And I know that there's probably so many people involved in all those surveys of those plots in the past and measuring the soil. So thank you to all those people too that we didn't get to talk to today. Thanks for interviewing and good luck in your future research.
thank you so much. Thanks for the invitation. And yes, thank to all the collaborators that allowed me actually to be part of their team, develop this nice work.
Great. Okay. Thanks,
Thanks.
Jose.